questions at the very end altogether, you know. So we come to our next presentation. This is by Professor Xavier Fernàndez Busquets. He is the head of the Nanomalaria Joint Unit at the Institute of Bioengineering of Catalonia in Barcelona, Spain. And he will talk about loading of antimalarial drugs into non-infected red blood cells.
Yeah, okay. Thank you very much, Reto, for the presentation. Because Professor Herling has already introduced the importance of malaria, I don't have to explain you how critical is this disease, particularly in Africa, so I can proceed to the next slide, which is actually to explain that our group, since the last 5 or 6 years, has been involved in trying to apply nanomedicine to malaria, and we have made an effort to show that nanomedicine is not necessarily something made of very expensive nanomachines, that we have nanotechnological tools like polymers, liposomes, etc., that can be used to fight malaria. And one of our personal commitments in the group is to contribute to this, to balance this wealth distribution inequality by developing nanotechnological tools to fight malaria. Then, as we have seen already before, there are 4 main legs to fight malaria. Vector control, vaccines, diagnosis tools, and drugs. And we are focused on the drug problem, which, as we have seen, is important because of the evolution of resistance by the parasite, which has been developing resistance against every single drug that has been used against it. And then because new tools probably will have to be needed continuously, because we must assume that any new drug that will be developed will suffer of the same problem, we believe that nanotechnology in the form of drug encapsulation can help to improve drug efficacy. Essentially, the objective of any nanotechnological approach based on targeted drug delivery of nanoparticles is exemplified here. On the right above, you have the way most of our antimalarial drugs are administered today, which ends up with the compound free in the blood for any cell to take it. And then the objective should be that one that you see below, trying to encapsulate the drugs in some type of targeted nanovector. That in our original prototype, we were very optimistic, and we designed a very fancy strategy where these nanovectors would be first directed to the infected red blood cell, if we are talking of blood stages. And then, because in this particular cell type, the parasite is inside the vacuole, and maybe some of the drugs need to target some Plasmodium organelles that are placed behind other membranes. We speculated that the nanovector would be very complicated, and that would make it very expensive. Actually, we realized that very soon when we went to the pharma companies who might be interested in developing this, and they tapped our shoulder and told us, come back when you have something much simpler and that it works without so many complicated steps. So this original Russian doll nanocarrier was simplified, and we have somehow tuned it down a little bit. Essentially then, our first prototype that I have shown here already a couple of years ago was an immunoliposome, where we were using a liposomal nanocarrier with specific antibodies against Plasmodium-infected red blood cells, and loaded with an antimalarial drug. For the first proof of concept, we substituted a fluorescent marker for the drug to see what was going on with the liposomal contents. And then, as you can see in this example, where we selected a field with several dozens of non-infected red blood cell and only one infected erythrocyte to show clearly what was the performance of our nanovector. As you can see on the above on the right, this is the targeting antibody, which targets only the infected red blood cells. The nuclei label tells us that that cell actually is the only infected one in the field, and the fluorescent quantum dots shows us that indeed the contents of the liposome can be injected into targeted cells and not into healthy erythrocytes. That was very nice for an in vitro assay, and we were too optimistic, thinking that we had already a magic bullet against malaria, and then we went to load, instead of quantum dots, antimalarial drugs and try this prototype in vivo. And then, as you can imagine, the result in vivo was not so good. We could improve significantly the efficacy of the free drug, either chloroquine, artemisinin derivatives, or primaquine, but we could never cure the animals. And then this was as good as nothing. And the conclusion was that we had a good first prototype, but the idea was to modify it in order to make it better. And then among the several different modifications that could lead us to use better drugs, maybe to target new stages of the parasite outside from the red blood cell, I am going to show you briefly our efforts in achieving a better targeting, better encapsulation, and a better in vivo efficacy of the immunoliposomal model. The liposomes maybe are not the best encapsulation method for malaria today, because we would need nanoparticles that could be formulated for oral administration. But nevertheless, we think that liposomes might be adequate for a future scenario of malaria eradication, where we might have very few cases of highly resistant Plasmodium strains. Then maybe in this case, a good immunoliposomal model could be useful. Then what we are doing is simply like playing a Lego game. We exchange the different parts of the nanovector to make it better. And the first thing we attempted was to achieve a better encapsulation. In our first prototype, the liposome was very leaky. It was made with unsaturated lipids, and we realized that when we were adding the liposome to the culture, most of the drug had already leaked from it. Then this is just to show, because these results were adapted from previous work by other laboratories, that it's relatively easy to achieve liposomes that do not leak the drug while in storage, or they don't leak it for several hours while in culture, simply by using saturated lipids and using pH gradient strategies where the drugs can be encapsulated with high efficacy. In this way, as I am showing below, we can now select between liposomes that release the drugs very quickly, or that contain the drug for a very long time, which, depending on the type of target cell, might be something that, or depending on the type of drug that you are using, you might want to choose between a rapid release or a fast or a slower release along several hours or even days. The next thing that we did is, I was not telling you all the truth when I was explaining the good efficacy of the antibody in the fluorescence microscopy, because that was achieved with extremely high antibody concentrations on the liposome, so high that it made that prototype absolutely useless in a real case scenario, because you could not afford to pay those liposomes for African regions. Then when we went down to amounts of antibody that we considered adequate, the efficacy of the targeting was not so good, as you can see here. The places on that histogram where you do not see a bar, one of them is this very good antibody that we were using that only worked at these very high concentrations on the liposomes. Therefore, we decided to try another strategy, which is, okay, it's very difficult to find target proteins for infected red blood cells, because the parasite has a tendency to change those exposed antigens very quickly, and they are highly variable. Then we went for a target protein which was in all red blood cells, and so we selected glycophorin A as a proof of concept, saying if we have a good antigen, that would be the results. Then, as you can see here, antibodies against glycophorin A, in this case, obviously, we're targeting all red blood cells, both infected and non-infected. And this showed, indeed, a very good efficacy in targeting all the cells, as you can see below. And also, it was something that we did not expect in the beginning, but that one has always to consider when designing medicines in which you have to use up your investment very well, is how many of those liposomes were used, because normally we only look at the cells. We want that all our cells are targeted, but we don't care if this job is being done only by 1% of the liposomes. In this case, we wanted to make a good use of most of our nanoparticles, and as you can see below on the right, where we show the retained liposomes with this one of our immunoliposomal models, all the liposomes were used for targeting the correct cells. So almost no liposomes were wasted. And actually, this gave us the expected result in vitro assays. This is the normal situation that we had before with antibodies against exposed antigens on infected red blood cells, which the parasite only starts showing late in the infectious cycle at trophozoite or Schizen stages. By targeting glycophorin A, obviously, we could enter the infected red blood cell much earlier. And this gave us an edge that indeed made that, for instance, chloroquine, the same concentration was much more active in our immunoliposomal formulation, both when added to ring stages and also to later stages than as a free drug. That was an important improvement. And we thought that would be the consequence of just entering infected red blood cell early on in the cycle. But then we realized that we might be witnessing here another phenomenon, which is because we are also loading the drug into non-infected erythrocytes, we might be using the erythrocyte, as it was already described some years ago, as carriers of drugs. Red blood cells are very good drug carriers because they are obviously biocompatible. They are eliminated by natural means. They have a very long blood residence time of up to 2 months. And then we guessed that what we were witnessing was this added effect. And indeed, when you load liposomes with, for instance, pyranine to see where the contents are going, we could confirm that we could inject pyranine not only in Plasmodium infected red blood cells, which are below, see the green color, but also non-infected red blood cells were receiving our liposomal contents. Then our hypothesis is that this is the classical way, our original prototype, where we were targeting liposomes only to infected erythrocytes, and then obviously in this way you kill the parasite that is in those cells. But Plasmodium, which escapes this strategy, is free to invade new red blood cells where they have a safe harbor, let's say. But what happens if Plasmodium, when it tries to invade a new red blood cell, finds the drug already inside, which is what we have with this model? Obviously, the parasite has much more complicated to survive in this situation, is what we call the Little Red Hood strategy, when Little Red Hood finds the wolf already inside. And indeed, when we were doing the same in vivo assay as I was showing you before, in this case, with this immunoliposomal model, we could eliminate completely the parasitemia from infected animals, and the animals could be cured. And I thank the two students who took most of this job, and I thank you very much for your attention.